I'll be updating on the progress we made on breeding coffee leaf rust resistance by crossing with cultivar and Obata. Okay, this is a project supported by SCRI USDA, and these are all the co-PI. And Dr. Chifumi Nagai is my predecessor, and she started the coffee breeding project at Hart since 1997. And in 1999, she's already crossed Katimua with a lot of commercial variety that's commonly grown in Hawaii. And but then since we did not have coffee leaf rust here, they just sitting in the field because we couldn't screen for resistance. And since 2022, I made new cross, trying to imitate what Chifumi did, but with Obata. So basically, this project is to screen the F2, the third generation of the Katimora crosses, and Obata F1, which is the second generation of Obata crosses. Good thing we have Lisa who developed the bioassay for CLR. We can check for their resistance in the lab, and also Da Peng in Maryland had developed a panel of molecular marker. We can verify what they are, and Qingyi will identify DNA marker associated with resistance. And we are doing field trial on 4 different farms at 4 different islands. And Tracy will be responsible for phenotyping, including cupping. And at any of these steps, Andrea will disseminate, and Matt will disseminate the information we generate from the project. So the project started in 2022. We focus on generating the seedlings from the crosses. And last year, we focused on the screening process, both the bioassay and DNA typing. And this year, we're focusing on installing the field trial. Okay, I showed a lot of these slides before, so I'm going to go through it fairly quick. We generate more than 3,000 F2 plants from the Katimora crosses. One of the first group is actually being in the field since early 2000s, and it's a cross between a mocha type, a mocha plant that was identified in Maui. MA stand for Maui, with one of the 2 Katimora we have. It was generated for genetic mapping purpose. And then the rest of the plants we generated in 2022. Unfortunately, after we did DNA typing, we had to throw away 3 different crosses, and I'll explain that later. Because they're not what we think they are. And similarly, I was trying to do cross Obata with the Hawaii commercials. So OA stand for Oahu. It was originally identified on Oahu. KA stand for Kauai, Maui, Kona. And I separate them into Mocha, Red Katrai, Typica, and Yellow Katrai group. And we also did some crosses between Obata and Ethiopian accession, which are nematode resistant. So we're trying to capture both nematode and CLR resistance. And unfortunately, 3 out of the 31 attempts we made are not successful, so we had to get rid of them later. And that we only identified that after we did the DNA typing. And half of this cross was also the self of the female parent. I believe I showed this slide last year, maybe. Anyway, for the CLR bioassay processes, we went into the forest in Monoweli. Our experiment station in Monoweli is surrounded by infected feral coffee tree, and we use that as our spore source for inoculation. And we started out using Lisa's method of making leaf disc and inoculate each disc with the spore. And you can see the Cona typica susceptible and develop beautiful spore and no spore on Catimora. But quickly we realized by doing these leaf discs, we will not be able to go through the 3,000 plant in a short period of time. So sorry, the last slide was just show you we incubate, oopsie doopsie. We need to incubate the inoculated disc for 4 to 6 weeks under condition that's suitable for COR, not necessary condition good for plants. You notice we cover the lights with shade cloths. Lisa has discovered that under strong light, the COR does not develop spores. Okay, back to what I was saying. We quickly realized doing leaf discs, we can't go through all those plants quickly. So we start playing with not cutting the leaf. We start out using the whole leaf. But we realized a lot of leaves start to decay. Can you see my cursor?
Yeah,
Yes.
we can see it.
Okay, so we realized leaf did start decay from about 1/3 this way to the petiole from the midrib, and then the decay spread from this point to the rest of the leaf. So we start cutting off the midrib, like half of the midrib, but we also realize the symptom develop much faster in this area. You see, it's more severe in the leaf area towards the petiole. That's why we start cutting leaf that look like this, and then just inoculate 16 drop each. And the result is we can still see nice symptom develop, spore development. And for a lot of them, they develop these spots, but no spore development. We call this tolerant. So this is susceptible, tolerant, and this is completely resistant. And at the end, these are the number of plants we identify from each group. Okay. And if you analyze the ratio between, if you lump the resistant and tolerant together, the ratio between that to the susceptible one, it depends on which type of crosses we're looking at. Red Catrine and Mocha, we get more of the resistant, more plants pass the bioassay, let's put it that way. And Typica has the fewest plants that pass the bioassay. Also, from the same cross, we use multiple individual, different F1 plants give us different ratio. The most significant effect is the maternal effect, meaning which plant you use as the female parent. It has a lot more effect on than the paternal, the one you use as pollen source, which means in the future we should use the resistant one, the Catimora or Obata, as the female parent. Unfortunately, I only had 2 Obata tree, so for all my Obata crosses, it was using the Obata was using as the pollen source. And overall, there's a 9-to-7 ratio, approximately 9-to-7 ratio between resistant, tolerant to susceptible. And it kind of indicate there are probably 2 major resistant loci that's required for the resistance or tolerance. So after we finish bioassay, the resistant tolerant cultivar F2 and all of the Obata F1 were sent to DNA typing. In all, we sent about almost 2,500 plants for genotyping. And the result, I'm just doing a very brief summary. Most of the F2 were what we think they are, except the 3 crosses I mentioned earlier. And 2 of those crosses, Shifumi actually shipped flower from Maui over to Kunia to use as the pollen source to pollinate onto 2 cultivar plants. So the cultivar were used as female. But it's most likely the pollen viability dropped. So almost everything is the selfing of the female parent, which makes sense. But the third one, even though it's a mocha plant that's used as a female parent, everything turned out to be pure cultivar, which we think it must be mixing of seeds. Otherwise, I could not explain it. So, and for Obata F1s, 3, as I mentioned earlier, 3 out of 31 attempts were the self of the female parents. 1 cross generate about half-half. But we have enough plants to go on. So right now, so the next step this year we're focusing on installing field trial. The first one was installed this is supposed to be 24. Last year, at the end of last year at Doe. And next week we're shipping to Maui. And for Kona, we already obtained the permit. We're waiting for the okay from the farmer. For Kauai coffee, we're still waiting for the permit, interstate import permit from HDOA. And then the leftover plants will all go to Tracy at P Park. So after the field trial is installed, we'll collect field performance data, and eventually we want to check the cupping quality, and we would like to use chemical analysis to predict the cupping quality to reduce the number of plants we need to cup. So this is when we first planted at Doe. I was surprised that they actually put cages on every plant. I never seen that before, but we do use that for cacao, but I've never seen that on coffee. And recently we started taking down the cages. And actually it took us too long to screen through the plant in the greenhouse. And they are actually already starting to flower. So we pick off all the flower buds to prevent it from the plant spending energy in reproduction instead of growing. Yeah. Okay, so because the plants selected from our project, they're still the genetics, they're still segregating, they're hybrids, so they had to be clonally propagated before we can distribute it if we select any plants out. I'm going just to mention a little bit on the progress we've done with the other SCRI project with Shack on the micropropagation of selected coffee cultivars. So Tracy kindly gave us 24 plates on Maite, which is identified in the growers fund, that's CLR-resistance. And we were propagating it in this bioreactor. Unfortunately, at the end of November last year, we have a massive contamination event. The air condition, the dehumidifier, everything broke down over the weekend. We came in to the lab on Monday. The autoclave room was covered in mold. we lost everything we had in the bioreactor. And only until recently we think we figure out we clean up all the contamination spores in the air. But right now we still working them on plates. We hope to start the bioreactor pretty soon. The bioreactors are good. You can multiply a lot in a very at once, but when things like this happen, you also lose a lot at once. So right now, the most advanced plants, that's what they look like, but we don't have many of them because of the contamination. We also working on the Fukunaga with Rasana, in order to distribute it to growers so they have seeds to grow their own rootstock. And we are also working on Obata, multiplying Obata. In addition to micropropagation in the lab, we also installed 2 small isolation plot of Obata. So hopefully we will be able to provide Obata seeds without having to bag the branches when they are mature enough. And these are the people involved in the project, and without them, nothing will happen. Thank you. I'll be happy to answer any question.